Welcome to the second part about the proteins, and today we are going to discuss their functions. There are many functions and we will cover most of them. So we will start with the enzymes, but until we explain them we need to do a little bit of chemistry. So for example, if we have the reaction of A plus B and we transform them into the C plus D, into these products, we can describe this reaction into the numerous ways. But one way is to describe their energy state. So on this graphic, on the y-axis we have the energy, and on the x-axis we have the direction of the reaction. As you can see here, energy represents how much energy these molecules contain, or as we say, the reaction states. So A plus B here has the higher energy than C plus D. This means that energy is spread into the heat as the reaction continues on. So this is called the exothermic energy. But even though that energy is being spread, it doesn't mean that it will proceed instantly. We still need to invest a small amount of energy so we can disrupt the bonds within A and B for them to create C and D. And this energy is called activation energy. So what does catalyzation mean? Catalyzation means to lower this activation energy so we can speed up reactions, which is very necessary for the biological organisms, because we would be practically dead before they would even finish. So enzymes do catalysis and they just lower activation energy. Now, how do they do it? Well, they create a microenvironment to effectively catalyze reactions. For example, we have the active site where this microenvironment is created, where we have ligands and other reactants coming through, mixed together, and the products are spread out. And they can also be regulated by non-competitive inhibitor sites and other mechanisms. This is the separate site to minimize the activity of the enzyme. Well, why do we need it? Well, we will explain throughout the video. So let's start with this scenario. We have a ligand, we have a product. So as you can see, when we take an aqueous solution of the ligand and we put enzymes inside of it, some ligands will eventually collide with the active site of the enzymes, and these enzymes will catalyze the reaction and create the products. But now, what if the biological organism does not want to create as much products because there's no need of it? Then we have to minimize the activity of enzymes, and that's what inhibitors do. We have two general types of inhibitors. The competitive inhibitor actually has the same or more larger affinity, so larger likeness, to attach to the active site of the enzyme. For example, we have this aqueous solution of competitive inhibitors and the ligands, and there's more likeliness that competitive inhibitor will attach to the enzyme and active site will be blocked for a certain amount of time. Now, non-competitive inhibitors, on the other hand, will attach from the other side of the protein, not in the active site, and they will change the conformation of the protein, structure of the protein, so that active site will no longer be recognizable by a ligand and reaction cannot be catalyzed. Well, this was about the enzymes. We'll continue with the structural proteins. So, for example, collagen, which is created from 3 alpha helices. Why do we need structural proteins? We need certain structures to be assembled inside of the cell and outside of the cell. For example, in biological organisms, collagen is mostly used as the major component of the connective tissue. Without that, we wouldn't be able to do practically anything, and it creates bones, tendons, it is used in muscle, and it generally connects everything. That's why it is called a connective tissue, and it gives us rigidity to do certain actions. Next is the transportation proteins. Well, there are generally many types of transportation proteins. So, for example, a glucose transporter, which can transport molecules from outside of the membrane to inside of the cell. So they will just get transported, usually by hydrolyzing ATP, so putting energy, or through facilated diffusion, or through other mechanisms. Next is the hemoglobin. Hemoglobin is the cell within this is actually a protein within the red cells of the blood, and they transport oxygen throughout to every single cell throughout the organism. And hemoglobin does that, so they transport oxygen, but they just move through the bloodstream. So that's the major difference between these two. Well, next are the storage proteins. What do storage proteins do? They store certain molecules. For example, in liver we have a ferritin, which is made up from 24 different subunits. But what do they do? They actually take a very toxic iron form and they just stuff that inside of them, so they actually store iron into more applicable form, so it can be later used to regulate iron levels into the blood, or maybe used to create proteins or other structures which need iron inside. Next are the receptors. Receptors will interact with the extracellular signals and change and basically give a signal inside of the cell to generate a response to that extracellular signal. For example, blood sugar is really high inside the blood. What happens? The insulin gets secreted a lot and insulin receptors get activated. Now, insulin receptors, through a very complicated mechanism, will activate glucose transporters and push the glucoses outside of the cell from the blood inside of the cell. So basically lowering down the blood sugar levels. And at last, and also we also have the messenger proteins. So for example, insulin over here is the messenger protein, which consists from the two different chains connected by the disulfide bonds. And what do messenger proteins do? They just deliver messages and they just give as signals to these receptors, so cells can respond appropriately. And at last we have the defensive proteins. The antibodies are created in response to antigens. Well, one of the kinds of defensive proteins are the antibodies, which consist from 2 heavy chains and 2 light chains. To describe structure more in details, well, we have the Fc domain. This is basically leg, not a very variable structure. And we also have the Fab domain. This is really variable structure because we need to identify and create antibodies for every single possible pathogens, and if that doesn't happen, the host will die. And at the top we have the antigen binding sites, where certain epitopes will get connected, and these have a very variable regions, so that it can detect practically every kind of pathogen. So, in conclusion, the proteins can catalyze reactions. Proteins can create structures within and outside of the cell. Proteins can transport or store different molecules, and proteins can act as messengers or receptors, and proteins can aid organisms to defend themselves from pathogens. Thanks for watching, and see you into the next video.